Hey everybody, welcome to our first exam review session for hematology. So we will be covering the PowerPoint that I sent out in the email. The exam for this week is going to cover weeks 1 through 3 material, so that's kind of what we'll go over in the PowerPoint as well. A little bit on the exam itself before we jump too far into it. It's going to be 43 points. Most of the questions are multiple choice, but there are some short answer type questions as well in there, but 43 total points for that. All right, so now with that being said, we will go ahead and I'm going to share my screen so we can look at the exam review PowerPoint. All right, so each one of you should have this. Just put this into slideshow. Yeah. Okay. All right, so we're going to go back to week 1, chapter 1. So I'm just going to do some big key highlighted takeaways from each chapter, but be sure to still go back, look at the actual lecture PowerPoints from those chapters, listen to any lecture recordings that you want to, and just kind of re-look at your actual material out of the textbook. But these are going to be the big kind of highlighted points out of each one. So in chapter 1, it was just a broad overview of hematology in general. So we learned the function of the 3 different cell lines. So of course the red cell function, which we've been learning more in depth on now, is that it's to carry oxygen to the tissues and also remove carbon dioxide from them. White cells is all about defense. They're there to help defend our body. They're a major part of our immune system. And platelets is blood clotting. So it's all about blood clotting for platelets. There was another task called the hematocrit that we've been learning that was kind of introduced in the broad overview in chapter 1 PowerPoint. But a hematocrit is also known as a packed red cell volume. So it's kind of, you spin the tube down and it will pack those red cells at the bottom. So it will really be the ratio of those packed red cells at the bottom to the entire sample with the plasma on top. So a hematocrit, also known as a packed cell volume. In chapters 2 and 3, this is just kind of more specimen collection, safety type stuff. Hand washing, always #1 way to prevent infection. It will break that chain of infection cycle. For accidental needle sticks, most of the time that we see an accidental needle stick happening is because the needle wasn't disposed of properly in the first place. So when you're in phlebotomy, you always learn that you immediately put that safety on and you immediately get it into the sharps. Still true, because that is the best way to get an accidental needle stick if you don't do that. And then for specimen actual tubes, in hematology, it's all about that lavender top EDTA tube. We use that for pretty much everything. And again, we are using a whole blood sample. We're not spinning it down. We usually get it and we put them on rockers in the hematology area, and they rock so that way they continue to mix. But we leave it typically whole blood, but again, they're in that lavender top tube. For coagulation, which again, we'll learn more on coagulation at the end of hematology too, this would be collected in a light blue sodium citrate tube. There is times that we do see that light blue tube used in hematology, not just in coag. And one of those times is when we have platelet satellitosis occurring. That's where platelets are kind of circling around the outside of the cell. And this happens when somebody's blood kind of reacts funny to the EDTA. So sometimes as a result, when you go to look at it in the microscope, you will see those platelets circling the cell. And when they run that through the analyzer, that analyzer doesn't pick up those individual platelets because it looks at it as one big cell. So the patient's platelet result is falsely low. So to correct that, once you see that in the microscope and you see the platelets are circling or satelliting around, we can redraw the sample into a sodium citrate light blue, rerun their platelet counts, multiply that result by 1.1, and then that's a better, more accurate platelet count for that patient. So that is one example of a good time that we do use light blue top in hematology world. Sometimes people's bodies will react funny with that EDTA tube. And then chapter 4 was all about the microscope. This is probably one of the first classes you're really getting to use the microscope. You'll be using it every week. So we're going to use it every week. So you'll get really good at it eventually here if you're not already. There was a few terms that you should know in regards to the microscopes. First term was resolution. This is just referring to the clarity of an object. So when you're looking in through the microscope, how clearly can you see the objects in there? So that would be resolution. It would be the ability to see two objects as two separate objects. They don't overlap, they're not fuzzy, that would be resolution. Parfocal is referring to a term when you switch from a low objective to a higher objective, you should only need the fine focus to bring it back into view. So we always start on our 10x objective, focus it. Once you have that focus, you're going to switch to either 40 or 100. And when you do that, you should only need that fine focus to slightly tweak it to bring it back into focus. So that's parfocal. And then there is a way to calculate total magnification, which you should know. That would be your ocular, your eyepiece lenses, times your objective lens. So that would be a total magnification. So in this case, if your ocular lens was 10x and you were using the 40x objective, you simply go 10 times 40, which is 400, and that would be your total magnification. Chapter 6 was in week 2. We do have a lecture recording of this for you to listen to if you'd like. It's still knowing the basic function of each part of the cell. You don't have to know it in depth. That was kind of what anatomy and physiology classes are for, but it's just knowing the basic functions of these parts. So overall, the cell is organized into 3 areas: membrane, nucleus, and cytoplasm. The membrane is just what helps keep the cell shape. The nucleus is considered the brain of the cell, and the cytoplasm has a ton of different organelles within it. With some of those organelles, they're listed here. All of them are listed back in the Chapter 6 lecture PowerPoint. You can go back and look at that. But mitochondria is a big one. I always just think energy right away when I hear mitochondria. The Golgi is what will package and sort molecules. Ribosomes will make RNA and proteins. Lysosomes are kind of viewed as granules in the cell, and they help in the phagocytosis process. The other thing I should mention real quick that I mentioned back in the week 2 lecture recording when I discussed Chapter 6 was that Golgi apparatus. That's that one that when you stain the cell and look at it under the microscope, on young cells that still have organelles, like most of our mature cells, they don't have organelles anymore, but if you go back to the really young cells, that Golgi will be viewed on there right next to the nucleus in the cytoplasm, and it doesn't take up stain. It's that whiter area that does not take up stain right next to the nucleus. So just keep that in mind. Go back to Chapter 6. I show a picture on it in that lecture recording and in that PowerPoint, so you can see it there. All right, Chapter 7 was learning all about hematopoiesis, which just means the development of our blood cells. So first we learned the fetal hematopoiesis timeline, which you still do need to know. So it all starts in the yolk sac, then it goes to the liver, and then to the bone marrow. There is a timeline process. So go back again to that Chapter 7 lecture PowerPoint. But roughly the yolk sac is for the first few weeks. The liver will take over 2nd month through the 5th month. And the bone marrow will really become the primary site in the 6th month. And again, that bone marrow is what stays our primary site after the 6th month on through adulthood. As far as other organs that are involved in different things, the liver can pick up and perform hematopoiesis if your bone marrow should ever shut down and fail. There are also cells within the liver called Kupffer cells that are macrophages, and they do a lot with helping get rid of debris and dying cells. Oh, that just got repeated right there at the top. Other organs, spleen. Spleen performs a lot for us, but mainly it just always is filtering blood. There's macrophages in the spleen as well, looking for dying cells, old cells, things that just shouldn't be there, that kind of thing. And then the spleen will also store a third of our platelets at any time. Thymus is all about T lymphocytes. That's what turns it into a T lymphocyte. So we make our lymphocytes in the bone marrow, and there's 2 types of lymphocytes. There's B and T. Overall, they're just made as lymphocytes. Then some of them go and travel into the thymus. When that happens, those that travel to the thymus will become differentiated into T lymphocytes. Those other lymphocytes that stay in the bone marrow become B lymphocytes. And then lymph nodes is where you will see the B lymphocytes hang out. So after the bone marrow has differentiated those lymphs into B lymphs, they will primarily hang out in the lymph node area. So the lymph is all about lymphocytes. That's why it's named that. Within chapter 7, we started looking at that whole hematopoiesis chart, and that's when we learned at the very top of that chart is what we have, our hematopoietic or pluripotential stem cell. And that stem cell can basically give rise to any cell out of the blood cells. So it can really become any cell. And upon doing all the studies and things like that that was in there, they learned 3 main properties of that stem cell. It's 1, capable of self-renewal. It can differentiate, again, into any cell, and it's able to reconstitute in an entire irradiated bone marrow. So if you were to irradiate a host, wipe their bone marrow clean, put in stem cells, they can reconstitute and build back up that bone marrow full of cells. In order to, though, get to these mature cells at the bottom, from the stem cell down to the mature, you have to have cytokines present along the way. These are also known as growth factors. So if you don't have them, you'll never have hematopoiesis fully occur. The cells will die off. So again, very important to help regulate differentiation and maturation of cells so that we can go from the stem cell down to the mature cells. So there's lots of different types of cytokines. There's a whole list in the Chapter 7 lecture PowerPoint. If you go to the actual chapter in your textbook, there's a ton of talk on this. So different types would be interleukins, GM-CSF, which targets just the granulocytes and monocyte line, erythropoietin, which is all about the red cell line, and thrombopoietin, which is all about the platelet line. So those are some good examples. And again, if you do not have these growth factors, these cytokines present, apoptosis will occur. And the definition on that apoptosis is programmed cell death. Your cells will be programmed to die because they're just sitting there. They can't mature, they can't do anything, so your body will kill them off. In Chapter 8 was our very first look into one of the cell lines, and so we started with the red cell line. I think next week, this week or next week, we start learning the white cell lines. I can't remember which one. But anyway, Chapter 8, all about the red cells. So here is the order of cells from the start, the hematopoietic stem cell, all the way down to the mature erythrocyte. You do need to know this order as it exists here. So again, it goes stem cell to myeloid stem cell to CFU-GEM, BFUE, CFUE. All of that is not visible to our eye. If we looked at it under a microscope, we could not identify those first 5 cells there. They're much too immature. But once you get to the pronormoblast stage, that is our very first identifiable cell under a microscope. So we can look at bone marrow and identify pronormoblast, basophilic normoblasts, polychromatic, orthochromic, reticulocyte, and erythrocytes. There are other naming systems, as you guys did in that study guide. Be sure you still know the other names for each one of those steps as well. So here is the overall characteristics of each cell. Keep these in mind because on this exam there are pictures. With the picture there will be a basic description because I don't expect you guys to be like total experts yet. We just started learning this. So there will be a picture but a basic description to go with it. So you should be able to, using this chart and some practice on pictures, which we're going to do here, you should be able to help identify each cell that is on there. So a pronormoblast, of course, is our biggest. It's 12 to 20 micrometers. Very large nucleus, 8:1 nucleus to cytoplasm ratio, and it does have a couple of nucleoli in it. Basophilic normoblast, a little bit smaller, maybe 0 to 1 nucleoli, but the key in this is it really gets a basophilic cytoplasm to it. It really gets dark and blue as it's making hemoglobin and stuff. Polychromatic, smaller yet, no nucleoli, and here the cytoplasm has kind of become a gray color. The other big thing with the polychromatic is inside the nucleus, it will start to really have condensing of the chromatin, and it's what they call you can call it a raspberry clumping. Like if you know what a raspberry looks like, they sometimes call the chromatin a raspberry clump. I always call it a checkerboard clumping. So to that nucleus, that chromatin in that nucleus is really key. Orthochromic, still a light gray kind of cytoplasm. It's getting lighter yet. The nucleus itself is extremely condensed in chromatin, a very dark condensed look. It's what we call a pycnotic nucleus. Reticulocyte has no nucleus. It's still larger than a regular erythrocyte and a little bit blue-gray because it has some remnant RNA in it. And then you finally have your mature erythrocyte, which is, of course, 8 to 8.5 micrometers. It has a nice central pallor to it. So when we look at some pictures, here's a few examples. Let's start with the top left. Up here, there's two of them, and they're the same cell. So when you look at this, first I see the cytoplasm color. It's gray. I look at the size of the cell compared to cells around it. It's bigger, but it's not like enormous. And then when you look at the nucleus chromatin, you can see it's clumping up in there. And this is the one that has kind of the clumps, yet patches, yet clumps, yet patches. That's what we call that checkerboard look. So these two would be the polychromatic normoblasts. When you go down, straight down to the bottom of that on the left here, that cell, if you look at the cells compared to it, it's almost the same size as the red cells around it. It's kind of pinker in cytoplasm, and then the nucleus is a solid dark mass. That's the orthochromic normoblast. That's usually the easier one for people to get. And then going over to the right, you got a big guy right here. Look at that cell compared to everything else. It's really large. So you know it's a large cell without even seeing the size of it. The nucleus, again, takes up almost the entire thing, dark blue cytoplasm. And I know it's hard to see in the picture, but there is definitely at least one nucleoli right here. Hopefully you guys can see my arrow. So this would be a pronormoblast. So I have another PowerPoint that we'll go through here right after I'm done with this one, and we'll practice again as it takes some time. All right, so when we learned on the red cell line, we learned all about the need for erythropoietin, or EPO for short. Erythropoietin is a hormone that's all about helping that red cell get from the stem cell to the BFUE down to the mature red cell. So erythropoietin, again, is made in the kidneys. It's got these cells in the kidneys that kind of sense when we're low in oxygen, when we have hypoxia occurring, and that will kick out erythropoietin, which goes to the bone marrow and helps make sure that we can mature red cells. It will let those red cells get released early. It helps shorten how long it takes to develop them, increases the amount of hemoglobin being made, all so that we can get more red cells out to the tissues to get the oxygen out there. There's another item that has to do with oxygen that I know that we didn't mention it yet in the Chapter 8 lecture, but I need you to know it, and I think it's crucial, so I'm mentioning it now. So please take it away. There's only one little piece to it. So there is a substance called 2,3-BPG. It's like bisphosphoglycerate, I think, if I remember correctly. I always just say BPG. But anyway, 2,3-BPG has a role in that the only job I need you to know it does is it helps oxygen get released from the hemoglobin and delivered to the tissues. So we know that the hemoglobin binds oxygen, the red cell carries and travels with it, and then that 2,3-BPG will help it release off, and so that oxygen can get to the tissues. That's it. That's all I need you to remember on 2,3-BPG. In Chapter 9 was all about different metabolic kind of processes. There is ways that the red cell has to get energy because, again, once it gets to a mature red cell, it has no nucleus, has no organelles, has a hard time making energy, so it needs something to do energy, and it does that through anaerobic glycolysis. This is the #1 way that it uses to get energy. It's also known as the Embden-Meyerhoff pathway. So at the end of that pathway, you'll get a total net gain of 2 ATP molecules. There is another pathway that's called the hexose monophosphate pathway, super important as well for red cells in that it helps give us glutathione. Glutathione is extremely important in protecting our red cells from oxidative injury. So those are the two pathways there that I really ask that you remember. All right, so not too bad. Again, those are just highlights from each chapter, big talking points. Be sure to go back into the actual lecture PowerPoint, listen to the recordings, because I always try to point out what you need to know in the recordings. So there could be more than just this. This is just a big, broad overview, key items. So now with that being said, I do want to practice a few more pictures since you are going to have pictures on your exam. So we are going to start with this first cell here, and I will send you this PowerPoint with the answer key so that you guys have it for practice as well. All right, so when I start here, I'm looking at the cell, and I'm looking again at the size comparison to those around it. It's a little bit bigger, but not a ton. Again, gray cytoplasm. And then when I look at the chromatin in the nucleus, I can really see dark areas that are condensed up, but I still see white patches. So that's, again, that checkerboard clumping. So this would be the polychromatic normoblast. I should also make sure to mention, and I know you guys have this in your study guide and it was filled in on the chart, the polychromatic normoblast is the last cell in the red cell line capable of mitosis. So I know that in that chart in the study guide it said, oh, pronormoblast, yes, cell division. Basophilic normoblast, yes, cell division. Polychromatic normoblast, yes, cell division. And then when you got to orthochromic, there was no cell division, and you guys answered no. So I want to make sure that I emphasize that polychromatic normoblast is the last stage capable of mitosis in the red cell line. All right. Now, this picture, when you look at it, large cell compared to the others, very dark blue cytoplasm. You can see the nucleus there, and within that nucleus, let's look for nucleoli. I see one, I see two. And I know they're not easy to see, but there are definitely two there. So that would be a pronormoblast. So that's a pronormoblast. Oh, this would be a good time for me to show you. Earlier I had talked about the Golgi not taking up stain being a white area next to the nucleus. Right here would be the Golgi apparatus. So it's that white area that just doesn't take up stain. All right. This cell, again, large cell compared to others, a bit more cytoplasm, still basophilic. Maybe one nucleoli, not much else. This would be a basophilic normoblast. And again, on your exam, there will be a size. There will be some details on the cell written out, so you don't have to use just your eye. You can use them together to help you determine. Okay, you can tell very basophilic cytoplasm here, big cell, again, a basophilic normoblast. Now this didn't stain well, everything's really pinky looking. It's not supposed to be pink, it's just the staining was really bad. But you can still identify it even with bad staining, because when you look at the size of the cell, it's a little bit bigger, but not terribly bigger. And again, the chromatin, you can see the dark and the light patches. So again, this is that polychromatic normoblast with the checkerboard clumping. Here we have a cell that's almost the same size as a mature red cell. The nucleus is a very dark matte chromatin condensation. So this is a orthochromic normoblast. That is what we call that pycnotic nucleus, where it's really dense. Here, we saw this picture earlier in our regular review PowerPoint, but again, this is that pronormoblast, very large cell, large nucleus, little cytoplasm. There is a couple nucleoli in there. The arrow down here pointing to this cell, it's about the same size as the red cells, dark, condensed nucleus, again, orthochromic normoblast. I should say if you go, no, I'm not going to wait on that. Never mind, ignore me. All right, these two same cells, very big. I know you don't have much to refer to the size of it, but really dark blue. And this would be a basophilic normoblast, both of these. This is a great example of an orthochromic normoblast, a dark, dark condensed nucleus. Here we go. And then this, bigger than the orthochromic that we identified here earlier, blue-gray cytoplasm, clumpy looking chromatin, so that's that polychromatic. Okay, so those are some examples. I'll send these PowerPoints to you guys so that you have them to refer to. The only cell I didn't really show you here is, obviously we know how to identify, I hope, a red cell. A red cell would be any of these cells sitting in here that have the nice central pallor in the inside. The only other cell that I didn't show is the reticulocyte. Now, not always reticulocytes are obvious on a peripheral blood. We all have them in our blood. About, again, 1 to 2% of our cells are reticulocytes. We have definitely reticulocytes and erythrocytes at all times in our bloodstream. But reticulocytes aren't always obvious when they're there. But again, the key features of a retic are they're a little bit bigger than the mature red cell. And again, there's still that slightly blue-gray coloring because they have remnant RNA left in them. So that's kind of a key on a reticulocyte look. So when you do your overall differentials and peripheral blood smears, you're just looking at the red cell morphology. But if I were to show you a picture of a reticulocyte, I would make sure that that picture showed a blue-gray tinge, and they're going to be a little bit bigger than the red cells. So that should be it for your exam review. I think you guys should do really well. If you do have any questions, please let me know. Otherwise, make sure you get an appointment in and have a proctoring time to get the test done by the end of this week. Thank you.